My research interests are understanding how different types of nerve cells are made in the spinal cord. So that's really the bit of your brain that runs down your back. And we're really interested in, as an embryo is growing, how do cells know that they should make nerve cells, and how do they know they should make particular types of nerve cells? And that's really important, because if we understand more about how nerve cells are originally made as an embryo is growing, we've got a better chance of being able to instruct stem cells to develop into particular types of nerve cells to help people who've got spinal cord injuries or various different neural disorders. We do a lot of molecular biology experiments and genetics experiments, which means that we're dealing with very small plastic tubes, and we're adding one type of liquid and taking it off and adding another type of liquid. And in the end, we usually end up with something to look at down a microscope, where particular genes are turned on or turned off in the zebrafish embryo as it's growing. And that we end up with quite beautiful things to look at, where every cell in the body that maybe has turned that gene on might be blue. And we can see that looking down the microscope.
I'm really interested in particular genes that we think are important for making special types of nerve cells. And the way we figure out what those genes do is to make mutations in them to stop them being functional. And the way that FISH helps us to do this is that they are transparent, so we can see the nerve cells develop in real time and real space, and we can use other tools as well, such as we can make different nerve cells fluorescent, and then we can ask whether those nerve cells change their properties as a result of the mutations.
I also teach a lab class, and we actually use zebrafish in those labs. So I think it's really important the students get a chance to see how vertebrate embryos develop, and so we actually show them embryos. We show them adult fish that have different phenotypes, and they get to actually work with the zebrafish, and a lot of them find that a really rewarding experience. So one of the things that attracted me to the biology department at ASU were the range of colleagues that are here. So I have some superb colleagues, and I very much wanted to be in a university department rather than, for example, a med school, because I actually really enjoy both teaching undergraduates and having undergrads do research projects in the lab.
It's wonderful. I mean, I had her as a professor as well my sophomore year, even though I joined the lab freshman year, so I got to know her before I even had her as a professor. She's been absolutely wonderful. She helps me with academics as well as everything in the lab, and she's always a resource to go to with anything I need on campus.
Syracuse is a great research university, and it pretty much came down to what I wanted to do. I wanted a good science program, so I feel like Syracuse was a great place.
What inspired me to first get into this area of research, and what still inspires me today, is the question of how you get from something that's as simple and as small as an egg, which is really just one cell, and sometimes it can be even completely symmetrical, and somehow that grows and develops into something that's as complicated as you or I, or in the case of a lot of my research, a zebrafish. And so there's all these little miracles that have to happen to make any type of living organism. And I wanted to understand how that happens, and I in particular wanted to really understand the role of genes in that, and that's really what I've spent my whole career doing.